Good afternoon, everyone. We're just going to wait a couple minutes while people get logged on. We have a little over 100 people registered today, and only about half that are logged on. So we're going to give it a few minutes before we get going.
$57. Everyone's enjoying the rain. Is it raining out in the valley? No.
Not today.
All right, everybody. We're going to go ahead and get started. There probably will be a few more that join us here in the next couple of minutes, but for the sake of time, we're going to go ahead and get started. Good morning and good afternoon, actually. Good afternoon, everyone, and thanks for joining us for the second installment of the Western Region CCA, call it Fall Webinar Series. My name's John Allan. I'm the Secretary for Western Region CCA. Today's webinar qualifies for 2 hours total, hour and a half of crop management and a half an hour of soil and water. Our agenda today consists of 3 speakers with substantial experience in their respective market segments. We're going to start with Dave Morgan, who will talk about fertility in grape production. Followed up by Sebastian Korob, who will discuss nutrient management in processing tomatoes. And then we'll finish up with Devin Clarke for a 30-minute presentation on soil health. So just again, thanks for being here. Just a couple business items that we need to attend to before we get started. One, I guess if there are any questions, please enter them in the Q&A box, not in the chat. We will visit each of those questions at the end of each presentation. This time, we are requiring that all attendees watch the webinar. We definitely appreciate those of you who are able to use your phone to call in, but in order to receive credit, you must actually view each of the presentations. So calling in isn't going to work this time. The QR code, it'll be shown at the end of the third presentation, which of course will be around 3 o'clock, probably a little bit before that. If you don't want to use the app/QR code, we'll be sending a attendee report to ICCA after the webinar is finished. As you all know, obviously it takes a little bit of time for that, for those hours to actually show up on your report. So we advocate using the app as often as you can, if you have access to it, of course. We'll post speaker contact information at the end of the presentation. That'll be in the chat. And I guess that's kind of it for the business side of things. So again, thanks for joining us. And we'll kick it off with Dave Morgan. Dave's the YaraVita product specialist for Yara North America. Dave, the floor is yours.
All right, thanks, John. All right, we should be good to go, John. I'm going to
Yeah,
assume so.
there we go. Perfect.
Okay, thank you. All right, yeah, just a little bit. My name, Dave Morgan again, both a PCA and a CCA, and we'll just get right into it. I found some pictures the other day that surprised me how long I've been doing this, over 30 years now. So last week it was very interesting listening to Paul Kraut and Jerome Peer and Danny. So this is my own presentation with some Yara information. I'm the king of Google and cut and paste. Sometimes to Devin's detriment, I don't always cite all my references, but generally I know where they're from. I've raised table and raisin grapes for professionally, I guess you could call it that, as well as been consulting on all types of grapes. But I'm not really a wine connoisseur, but we're going to kind of lean towards wine or just grapes in general. So there we go. Just my only shameless plug. Today we all work for Yara. Just so most of you know, founded in 1905 in Norway. I've been in California since World War II. First in the world to manufacture synthetic nitrogen. That's quite an honor. BASF and the Haber-Bosch process. We started with another process, and then we transitioned in the early 1910, 1912, 1915 into Haber-Bosch. Now worldwide presence. For the older generation like me, you know, you're going to remember Yara as the Viking ship brand, that logo, or the calcium nitrate company. And originally, Yara was formed to prevent and reduce world hunger. And then a whole lot more. And Devin can get more into that later if he so wishes. So this is interesting because in grapes, a focus might be in bottles, it might be in barrels, it might be in bricks, it might be in tons, how your vines grow. But basically, I'm going to call it the 5 R's. We know the 4 R's, and we'll talk about that intermingled today: source, rate, time, and place. But also, it needs to be successful for the grower for that return on investment. And I also love slides, so I'm a fan of more is more, as opposed to some people like less is more. So I'm just going to have a bunch of slides we're just going to go through. I'm not going to try to read them for you. You guys are educated and informed on your own. But basically, table grapes is a finished product. In fact, I just ate some for lunch, some late season Costco grapes in a clamshell, as opposed to wine grapes, which are a raw ingredient in a recipe. And so they're used quite differently, yet they're grown very, very similarly. You know, the old size matters or size doesn't matter. You know, obviously table grapes, you want to be big, high pulp. It says low sugar on this, where it came from, but you want medium to high sugar if you can. Generally thinner skins on some of the varieties, but some of the later ones now seem to be a little thicker. Generally seedless or seed trace or very small seeds, other than like Red Globe and some other older varieties, and generally low acidity. Whereas that wine grape berry, much smaller in size, lower in pulp, high sugars, thick skins, mini seeds and large seeds, and that higher acidity. When I first, because I didn't even grow up in a farm. I grew up in Fresno, so I had no clue what terroir was, and I used to kind of not really believe in it, but it does make sense. And basically, you know, that extreme conditions concept versus in that first paragraph versus the second paragraph. Most of my career, I've always done balanced nutrient programs, looking at vine physiology, nutrient removal, nutrient demand, for uniformity of crop harvest, crop growth, all of those things. This is a study that Yara did, and it's very fascinating. And you're going to say, how does this apply? But so Yara did a 5-year, 4-harvest variety test, treatment, demo plots, call it what you want. And they used all these different characteristics of coffee. And you see them in the little yellow circles. citrus fruit, acidity, astringency, intensity, bitter, nutty, chocolate, roasted, sweet, body, balance, and clean cup. And each of the blue dots were different fertilizer treatments, with and without calcium, with and without potassium, with and without boron, and then different forms of nitrogen. And you see how the fertilizer had an effect on the flavor characteristics of the coffee. And in fact, I just saw the other day, Yara signed a large agreement with a large coffee distributor manufacturer in Central America, in basically fertilizer and fertilizer relationships. So my point here is that that the terroir is correct, not just the soil, but how you treat them with your inputs. And obviously this is more of the wine grapes, and I won't even try to get into that, but just every variety has its own characteristics. But having said that, you need to look at what the vine has grown in, which is the soil. And I'm not going to get into soil. I actually have my bachelor's in soil science, one of the few and far between people. But basically you've got this is not exactly how they phrase it, but the physical properties of soil, the chemical properties of soil, and the biological properties of soil. Not going to go into a lot of detail. You guys pretty much know what these are. So we'll move on. Basically, you need to start with the soil analysis. And you kind of farm with the soil you have. That, people use sayings on soils and how much you pay for them, but every soil is different. But most of the time in that yellow statement, most soils, you need some sort of a long-term amendment plan, and then like a short-term fertility plan. Long-term amendments might be pH, EC, deficiencies, and short-term would be basically the crop removal and the crop demand. And we'll get into that here shortly. My 2 cents on the soils, though, things that are, and everyone's debatable, which are the most important things that a test tells you and shows you. But I like to refer to pH as like that restrictive point in the hourglass. You could have a soil full of nutrients, balanced completely, but pH is too high or too low, it's going to cause that restriction on that availability to the crop plant. And then the EC is the next one. How dilute is your EC? How, and how much like iced tea, how much tea is in it, how much sugar, how much ice, how much lemon. And a low EC is very weak, meaning no or very little elements are in solution, whereas a high EC is a very hot mix. And last week, Jerome did a great job talking about salinity and EC values. So looking at your soil, you can tell a lot. I can tell a lot very, very quickly looking at those 2 things. This is a really simple question, but when you understand the answers, it really gives you a foundation to build crop programs, to understand what the soil needs and what the soil supplies and what the plant needs. And so basically it's a very simple answer. There's 3 answers. 1, we provide nutrients not found in the soil. So from that soil test, we see those things which are deficient, whether that's in a CEC level, a ppm level, mill equivalents per liter level, however you analyze them, but nutrients are deficient. In the middle, we replace the nutrients that were removed from the soil from our previous crop and previous harvest yields. So we know that, like in the case of almonds, 68 pounds of nitrogen is removed per every 1,000 pounds of crop nuts, meat nuts. And so that's a number that we know is removed. And then the third one is balancing the nutrient demands, the nutrient requirements of that current or your present crop. So knowing that the plant is going to have a nutrient demand, and later on in this, we'll look at some of these nutrient curves and take it from there. Just a 5-second description. Basically, what makes plants essential? They need to let a plant complete its life cycle. You can't substitute it for something else, and it has to be over a wide variety of species. And over time, we see how these have been added to. There was these acronyms that most of us memorized, and now we've added cobalt. There was the iodine here a month or so ago, and also silica in some crops and in some different ways. So these are the macro in what we consider primary and secondary elements, and then the micronutrients. A lot of us don't. We kind of forget Botany 101, but basically the other big 3 elements that we don't pay for, carbon, hydrogen, and oxygen. And so as you see that first little diagram there, basically CO2, H2O in the presence of sun, chlorophyll, enzymes, we create sugars, and the plant gives off oxygen. And then in the bottom curve, it's and then that's photosynthesis. And then respiration is just taking that, those glucose sugars that are made and then breaking them down into plant growth. This is kind of a nice little graph, because as we go into these curves on plant growth, photosynthesis is that carbohydrate accumulation as we're growing canopy, but also we're depleting our carbohydrates at certain times of the year. And so it's kind of like the gas tank versus the engine. Growing up, my brother had a 68 Dodge Charger with that 440 Hemi in it, and that was the coolest car. That was one big engine, but it took a big gas tank to run it. So photosynthesis and respiration are basically just a balancing act that the plant does that we don't even see, we don't even hardly acknowledge, but it all takes place on that very cellular level. So basically, we're now going to go into the essential elements and talk about what their roles are, and then start building on from there. And really, if we understand the role of the elements, then we can make programs, we can look at products that are necessary, call them fertilizers, foliars, fertigation, soil amendments, additives, to try to reach whatever our crop goals are. The first element, and we're just going to go in order of the typical list, is nitrogen. You know, basically, if I said, what is the role of nitrogen? You're all going to say it makes plants grow. So it's found in proteins, chlorophyll, genetic material. My only comment here is most plants prefer the nitrate form. Plants like blueberries and plants like pecan trees generally can be more ammonium favored, but generally most crop plants prefer the nitrate form, although they will take up the ammonium, but preference. Deficiencies of nitrogen may or may not be very common in grapes, but if they are, you're going to see that reduced vigor, smaller shoots, smaller leaves, but basically a pale green color. That last comment, base your nitrogen on, you know, how the vine grows. Are you happy with canes, shoots, spurs, diameter, thickness, yields, your tissue analysis, all of those things, your crop removal. And we'll get into that. Phosphorus. Generally, I look at phosphorus like that Energizer bunny or that Monster, you know, energy drink. Phosphorus is the energy component in a plant. It has a lot of roles, does a lot of things, but basically you need more phosphorus. That P plus ADP equals ATP. So that P, I could make a little I. I'm not really that smart in all the computer functions, but it takes inorganic phosphorus inputs to add to ADP to make ATP, and then that just cycle goes round and round and round. but it does take inputs of phosphorus and which are sometimes lacking in our programs. I'm gonna make the comment now, I'm gonna talk about it later, but basically, if we're low in elements such as phosphorus, reactions either will take place slower, not last as long, or not to the greatest intensity that they could or should be doing. Phosphorus deficiencies, normally in most crop plants, you're going to see a purplish color. And it's interesting because you're thinking, you know, it does all these roles, but it doesn't really deal with photosynthesis. But because there is a magnesium phosphorus relationship, when phosphorus is deficient, that magnesium and those chlorophyll molecules are not able to function correctly. Wine grapes have a lot of viruses and a lot of could be insects, and so sometimes it can be confusing. Some of these colors that our leaves are going to demonstrate that may or may not be phosphorus deficiency, and that's where a tissue analysis helps you out. Potassium, most everyone has a pretty good handle on potassium. It's that water balance regulator in the plant, helps make sugars, transport sugars, improves our crop quality, our flavors, our colors, enzyme activation. You know, potassium is not found in any one structure, but it's in about 30 or more different reactions. So very important with potassium. Having said that, when we look at these crop demand curves, all of a sudden now we look at the timing of these inputs, how important they are. Potassium deficiency, basically that marginal chlorosis or necrosis. When I was just first starting out, you'd go look at vineyards like this and think, is this a deficiency or a toxicity? And so sometimes it can be confusing when those margins of the leaves, the very outsides of the leaves, because the potassium is basically sucked out or drawn away from the outside and transported to other areas, mainly the crop, we get that desiccation. And obviously low potassium is going to affect our cluster size, our fruit size, and then our ripening. And that's the other big uniform thing with grapes is uniform ripening. Calcium is not always a very easily seen deficiency, but it's necessary at every growing meristem and for root growth. I'm not going to go into that, but you know, we need calcium presence in the soil and soil solution for roots to grow. And calcium is only taken up in that brand new root growth, that first 1 or 2 inches of the root hair growth. And so we need active roots to take up calcium, and we need calcium to actually make roots grow. So also calcium, as you know, in that cell wall, middle lamella, cell membrane, strength, integrity. Calcium also works with potassium in that turgor activity with the stomata. And then it does play a role in the soil on a nitrate uptake. Calcium nitrate, calcium, the cation, nitrate, the anion uptake together. Deficiencies, like I say, more of disease susceptibility, weaker skins. I'm not sure where that picture came from. If you'd ever see a calcium deficiency, typically it's not a deficiency. I do have a few slides now that are kind of interesting. I'm going to go into that, but basically in a leaf, when that fungal spore tries to germinate, if it comes into a high concentration of calcium, the calcium will inhibit or reduce that botrytis type infection. And so the more calcium content you have in a leaf, the greater fungi resistance you'll have. This is a foliar work, and actually I have referenced it from Marshner. So this was that the first column is the days after veraison, call it veraison or berry change or color change, berry softening. Okay, the second column is control without calcium, without foliar calcium. So the percent of infected berries. So after 21 days, after veraison, you've got about 14% infected berries. And then the next two columns, the calcium sprayed post veraison or at berry softening, at color change. You don't get that same 14% damage until basically day 36. So there is some value of a foliar applied calcium later, after cell division, berry set, berry sizing. But if you look at the far right column, calcium applied at greenberry pre-veraison, that 14% damage is not really found until, say, day 45 after veraison. And so what you're gaining is many, many weeks of basically control based on that increasing the calcium content in that leaf. And also interesting to note here at the very bottom on the right, that 22%, that was the maximum at this study that the botrytis occurred, whereas that was the same beginning number at basically 31 days without calcium. And so you could go from 31 days to 70 and not have that increase in botrytis with foliar calcium. Not a plug for my calcium, but just a plug for calcium in general. This is something I found the other day, and I just wanted to plug it in. And most you guys that are actively in the PCA world making recommendations, this is not a recommendation or supporting anybody, but it showed the numbers of applications that get to be made. And even if you just did dusting sulfur, that would be different. But any fungicides, how often these are sprayed. So there's lots of opportunities for foliar nutrients to be applied, such as calcium. Magnesium's our next element as we're going through that central element, central molecule in chlorophyll. Chlorophyll photosynthesis enzymes that role with phosphorus, carbohydrate production. Very, very, very important element is magnesium. Magnesium deficiency, these are going to be much more common. Certain varieties, certain soils, you're going to see that very classic visual symptom. Some call it a Christmas tree. If the leaf is turned the right way, maybe you can see it. But basically, the veins are green, but then the green extends farther out than, say, an iron, which we'll look at. And then we have the margins are chloride. The second thing is you'll get that bunch stem necrosis, basically stems, petioles, rachis, our green tissue. And so without good magnesium content, we'll get stem necrosis. And this slide here demonstrates that this was from a few years ago, but it's still true today. If you have certain varieties on certain soils that, and maybe you, like I say, if you got table grapes you're holding, or if you've got grapes that go into cold storage, or wine grapes that are held on the vine a long time, and you end up with that stem necrosis issue, here's magnesium treatments, and you see you can reduce your stem necrosis from 2 times to 10 times a reduction in bunch stem necrosis. So again, another plug for magnesium that may not be appreciated in some of our daily treatments and fertilizer opportunities. Sulfur is not really a element we talk about in grapes, but it's an important, usually we use it as a fungicide, but elementally nitrate conversion into amino acids, sulfur is in some of those reactions and those molecules and compounds. Plays roles in vitamins, enzymes, chlorophyll. So it's an essential element. And this picture is a great example. It's kind of fuzzy, but basically sulfur deficiency is going to have, similar to a nitrogen, kind of a green, pale, uniform chlorosis, pale color. Probably not very typical, but under certain soil conditions or certain vines or rootstocks, perhaps. Now we're kind of going to switch from the macro to the micronutrients, but this was just something I wanted to share. This chart shows both the soil texture on the left and then the pH going across. So different soil textures typically have certain micronutrient deficiencies associated with that, basically due to low CEC and low organic matter content. So you go to the far right, pH is above 7, sandy soil, you could probably anticipate manganese, zinc, boron, copper, and iron deficiencies. And so knowing what your soil pH is, again, and your CEC and your texture is going to get you a good idea of what to anticipate for correction. And again, this is kind of how the micronutrients, when they act as these enzymes or these catalysts, but basically, you know, in that lock and key mechanism, they combine to make processes. And again, like I say, most of the micronutrients, along with the magnesiums and the phosphorus, you know, your reactions are going to take place faster, they're going to last longer, and of greater magnitude when everything's in a correct balance. Iron. Typically, we consider iron as photosynthesis element, works in chloroplast and chlorophyll formation, does a lot in the energy for these cytochrome, these ferredoxins. Some of you remember those names. I remember them, but they're long gone from my memory. But an important element in the leaf. Generally, it's that green vein. Solely the vein is green, and then a creamy or a white rest of the leaf blade. And then under severe conditions, normally, I will say this, when you get a deficiency of one element, generally you're going to have deficiencies of other elements. So when you see reduced growth, smaller leaf size, it might be combinations of, say, zinc or other elements. But typically that green vein, the interveinal chlorosis. I threw that little heme and a plant chlorophyll thing in there. Iron in our blood is very similar to the chlorophyll molecule. Iron in our blood and in plants carries that oxygen, and in plants and in humans, and then in chlorophyll, the magnesium is that central element. So very interesting how those are very, very similar. Zinc is a really important element in grapes, in trees, plants, a precursor to the hormone auxin, its production, its metabolism. It's an enzyme catalyst. It does a lot with a lot of different things. Fruit size, fruit set, yield, root development. Zinc is probably underappreciated as a fertigation element for root growth, but just as growing leaves and shoots is growing roots in the soil also. Zinc deficiencies, typically we see them in the leaf size, those shortened internodes, that rosetting of the leaves, also distorted or mottled apical leaves, the size, the shape. Zinc can be confusing with boron, but basically you've got a fairly decent berry set. So the berries have set, but you've got that shot berry, that big berry, little berry, as opposed to the, we'll get to boron in a minute. So, but basically the leaf internode, the diameter, and the leaf size, and then looking at the cluster itself. And then obviously tissue analysis is going to help you out. Manganese. We don't talk a lot about manganese, but it's, again, critical in chlorophyll, photosynthesis, genetic material, lots of other things. Some people talk about manganese in terms of seed set and other things of that nature. I won't even try to get into that. Manganese deficiency is more diffused. similar to the magnesium, but it's different. In citrus, it'll be a very chlorotic, but very not distinct in its deficiency look, very diffused. So you are going to have that chlorosis between the veins, but it's going to be more, it's not going to be as clear and distinct a line, if that makes sense. But when you see it in citrus leaves, then you'll know that they're very mottled look. Copper. Normally we talk about copper as fungicides, but copper is a catalyst in photosynthesis, respiration, all these other processes. In other crops, it works in pollen formation, viability of pollen, cell fertilization, which again, even though grapes are self-fertile, they do need pollen, or they do need copper, excuse me. Also plays a role in the lignification of cells and cell walls. And so something to consider. Generally, we don't see copper deficiencies, but if we did, they would be shortened internodes, pale color, and more distorted shoots. Boron. Sometimes we just talk about boron as just being talking about pollination, but boron has a lot to do with hormones, sugars, nitrogen, calcium, cell wall integrity, and then pollen viability. And this gets a little confusing. I threw all these pictures in, and then I do have a quote from the bottom. This was from Pete Christensen and Bill Peacock, farm advisors here in the San Joaquin Valley, now retired. But I'll basically read it. A boron deficient Thompson seedless cluster in the trial vineyard showed reduced fruit set, the presence of numerous pumpkin-shaped shot berries, and necrosis of some branching. Fewer than 10% of the berries are normal size and shape. So when we get clusters that are very, such as that one that's right above it with the black background, that large pumpkin-shaped berry, and only 10% are normal size, whereas zinc, you're going to have more normal sized berries and just small berries, whereas a boron deficiency, you're going to have that shot berry, that large oversized pumpkin berry, and then the small berries, because boron playing that role in pollination, there's just not enough boron to stick those berries. Now in table grapes, historically, you wanted enough boron to set the crop, but not enough boron to set all the berries. So you wanted to shatter off some of that crop. And that's why those early jib sprays were so critical. And that's why some people were paid the big money to make that call. Now a lot of the table grapes don't require that jib shatter spray so much. Boron deficiency, there's that one little picture with the red print, because it plays that role in calcium in terminal growth. If you are boron deficient, you'll see the terminal shoots just will die. Boron toxicity is a little more common in some areas, and you'll see much more rounded leaves instead of that same angular shape typical. So a lot depends on your soil and your areas. Some soils are deficient in boron, say the east side of the valley. Some soils are very high in boron, such as the west side, whatever their formation was on that. Molybdenum's, you're thinking, Dave, why are you talking about molybdenum in grapes? But molybdenum is important in the nitrate nitrogen enzyme process and in amino acids and everything else. Plus a role in pollen. And so what's interesting is, and I'll say this here, there's a disorder in Merlot, and typically it's been in Australia, but in that fine print, that was from the Yara North America or the Yara Australia website. But young vines with high levels of nitrate in the tissue often die or remain unproductive throughout the life of the vine. This disorder, known as Merlot disorder, and the symptoms are smaller leaves, leaf edge burn, poor leaf color, and the wood fails to mature. But basically, you don't get crop. okay, is the problem. And it's that high nitrate. So understanding that molybdenum affects that nitrate conversion in the vine. If you have any, when you do your leaf tissue analysis, if you're doing ppm nitrate, which I recommend in your petioles, and then total leaf nitrogen in the leaf blades. But if you get high nitrate nitrogen, and you're not getting good fruit set, berry set, you might want to look at molybdenum. So in this graph here, the control had, whatever the value is on the side doesn't matter, but the control had the disorder expressed. So when you added boron, it did not change the expression. So adding boron to this disorder doesn't fix it. And then they also tried magnesium, boron, and zinc, going back to why the role that boron plays and zinc plays in pollination and fruit set. It still didn't have any effect compared to the control. And then when they applied molybdenum, your disorder went basically down to 0. And so knowing what the elements are, that was one of the first slides, knowing what the elements do helps you can make corrections based on what you see out in the field. So boron or molybdenum moly might be really important in some of your varieties that are not getting good fruit set. Cobalt, cobalt and moly have come into designer nutrient status here the last 5 or 10 years. Enzyme catalyst component, mitochondrial respiration, again, taking that sugar and making energy out of it, nitrogen fixation in some crops, vitamin B12, anti-ethylene, and an anti-senescence effect, and also plays a role in lignin in your cell wall. We noticed lignin earlier with copper. So sometimes when we're talking about maybe grapes that are hanging a long time, or that this anti-ethylene, anti-senescence effect, maybe we want to consider a little bit of foliar cobalt into those post-veraison sprays if those grapes are going to hang a long time. Cobalt's rarely observed in grapes, but because it has that role in the nitrogen and these other processes, you'll just see a stunted or an oddly reduced growth rates. In seed crops, you're going to see differences in germination and legumes also. Nickel, I think we're about done with the elements here. Nickel's one of my favorites because it's in that urease enzyme. So plants converting ammonium and nitrate into these amino acids need the element nickel. Most commonly deficient in pecans, California, Arizona, New Mexico, Texas, the South. Basically, the deficiency of nickel is what they call little leaf or mouse ear. Pecan leaves should look similar to a peach or an almond, narrow and long, but when they're deficient in nickel, they're much more like little Mickey Mouse ears. But what happens is because pecans, they put on a ton of zinc. They're doing zinc sprays all the time, every year, time after time. And there's a relationship that the high levels of zinc interferes with the nickel background level. And so when you do get that deficiency, treatments of nickel will correct it. So it's kind of an interesting element. Chlorine, a lot of people talk about chlorine as issues, but basically it's important in photosynthesis and in these water splitting reactions. It's an osmotic element along with potassium. Again, that calcium or that cation anion balance. Potassium, the cation, the anion would be the chlorine. Role in disease suppression, also nutrient transport. So still an efficient element. Like I say, rarely observed in grapes is a deficiency. Normally it's a toxicity, as Jerome talked about, with high EC, saline soils, poor irrigation water quality, those things. All right, take a breath. Now that we understood each element, where do we go? Okay. So basically the merry, quite contrary. How does your garden grow? Well, how do your grapevines grow? So this was basically from Winkler at UC. The Iowa State had a pretty picture, so I put it in here. But basically, this is how grape grows. Don't really worry about the dates. The dates are probably for Iowa. So our dates in California, depending on if you're in the coast or in the valley, might be different. But the curves are what I'm going to talk about here. So the top, the blue, is that carbohydrate reserve. So carbohydrate reserved are the highest in the winter. And as the plant starts to grow, the carbohydrates are depleted, and that downward curve. At the same time, you have the shoot growth, that lime green. As your shoots are growing in your growing canopy, for a while, the canopy is consuming the energy, the carbohydrates that the plant stored. So once you get your peak of the canopy, is basically the bottom of your, you've used up all your carbohydrates, and now your canopy is set, and now you start to feed your carbohydrate reserves, and now you see the canopy going down and the carbohydrate reserves going up. And then kind of that light purple line, the berry size after bloom, berry set, then we get kind of a double, not a straight line, but a quick cell division, and then a cell expansion steady from there. And then right behind that is that red line of the bricks. So the bricks is going to start to increase very slowly at first, and then increase steadily up until harvest. Obviously, temperature conditions are going to affect, really hot days, really cold days are going to change how sugar accumulates. Crop load, a bunch of other things are going to influence that, but that's for another day. Tissue analysis, really important. That's how the good thing about grapes is you're going to be able to do foliar or fertigation applications, and you'll be able to tell by your bloom sample what you've done, and then taking either another sample at veraison or at harvest to get you many snapshots of what's going on. So building your program based on historical tissues as well as anticipated things. Also, I'm not going to get into it a lot, but down here, these ratios, some people really believe a lot of them. Some people don't look at them much, but there's a lot for how these elements relate to each other, as well as just the individual numbers. A lot of times, and also these colored bars and these colors, they may not be. They're going to tell you that this vine is basically sufficient, high in a couple elements and then low in some. But when you look at the raw numbers themselves, you've got to make sure that you're going to be happy with those numbers. This came out of Oregon. A lot of it was always based on the UC data. Paul talked about it last week. Some managers pull petioles, some pull leaves, some pull separate, one or the other. Sometimes when I'm out doing it, I will pull both the petiole and the leaf blade. On the petiole run complete, and your ppm nitrate, and then on the leaf blade is your percent nitrogen, and run complete. Years ago, there was labs I knew that would do like NPK and zinc. And I would say to myself, when a grower would show me those analysis, it's like, well, what about the other elements? There's a lot more than NPK and zinc. And we're gonna, and by going through the elements like we just did, you're gonna have a pretty good idea of why we need to look at these other elements. And the other thing is, and I make the comment, the petiole is the highway, but the leaf is the destination. But there's a time where the leaf is not the destination, the cluster becomes the destination, and that's why that potassium deficiency, everything leaves the leaf blades and they go to the bunch. So this was something from Joe Trainer. I do quote Joe on this one, and he talks about most of the standards were developed years ago, and they were developed on Thomson. And so even though there's been more analysis, and I'm not familiar with a lot of things, you really need to set your own standards in your own vineyards, based on your varieties, your rootstocks, your soil, your locations, and set your own standards. Find your best vines, figure out why they're the best. Find your worst vines and try to figure out why they're not, the best. But you'll be able to do that. And if not, we can help you out. Real quick sampling, that full bloom or early bloom sample, you'll be opposite the cluster. But then in that midsummer, that veraison, or even at harvest, you're going to be leaves 5, 6, and 7. Basically, that most fully expanded mature leaf. If they start cutting canes, if they start pulling leaves, then sometimes it's kind of hard to. You just got to kind of walk more in the vineyards. This was a typical. Some labs have presented that, knowing that the typical flow of the nutrients over the season gets you a pretty good idea of the demand. I'm going to keep moving. I'm going to run out of time if I'm not careful. This was talking about those arrows indicate the direction of the flow and also the size of the arrow. So early on, based on just like that other graph we saw shortly, all of your early bloom or your early bud break, everything is going out, growing canopy. And then until bloom, that B, 3 to 8 weeks until bloom after bud break, then we start getting photosynthates coming back to the vine, as well as going out to the new canopy growth. And then that C, the fruit set until veraison, now you've got things coming to the berries, going back and then going out, and then the veraison until maturity. Now most of it's going back to the crop, and then after harvest, everything goes back to the mother vine. This is something that Paul talked about. Basically, your nutrient removal, average removal, NP and K, high levels, low levels, but knowing what your what these are is going to make a difference. This is something I found from the Helena website. It said for every ton of grapes, it removes 8 pounds of N, 3 pounds of P, and 13 pounds of K. But really, it's looking at your analysis here, looking at your crop, your yield, and how do the vines look? How did they grow? To really put it all together. This is where you take the science and the art, and you got to marry them together. This was just a chart I kind of put together. So this was in those conservative numbers. So 1 ton is 3 pounds of N, a little over half a pound of P, over 5 pounds of K, 4 tons, 9 tons. But that's just the nutrient removal. So you've got the plus the vine use. So the vine itself, canes, spurs, roots, trunk, it takes a certain amount of nitrogen. And I just found these numbers somewhere. It's going to need about 20 pounds of N itself, with P and K and the other elements. And then there's the efficiency factor, because not 100% of what we put on, unless it's 100% foliar, which may not be possible in some crops in some areas, but that efficiency factor. So taking our nutrient removal in tons, taking our vine use, and taking our efficiency factor is going to get you how much elements to begin with your program. Similar to that first chart, basically you in the nitrogen, your greatest demand is from bud break until bloom, and then there's a little during berry fill or berry set and berry sizing. Okay, I've got a chart here coming up next on the right. So basically 50% of your nitrogen is going to be required in that pre-bloom stage, 25% post-bloom, and then to some people 25% post-harvest. Whereas if you look at the potassium, the K number, you need about 35% pre-bloom, but then you look at that percent, and if you look at that top red, that bunches category really demonstrates the potassium draw, the potassium demand is on that bunches. Same thing here. So early calcium when you're growing your canopy, potassium and nitrogen peak. Calcium does peak after berry set. Then you got your nitrogen and your potassium peak from veraison until we get to harvest. And then your magnesium and your phosphorus are fairly steady throughout the whole year. Real quick, I'm going to run out of time, so that's okay. So we talked about the elements individually. We test them individually, but they work together. And this is just an example of one chart. So you see all those green Y's are all the elements that are involved in these different things here. I'm gonna go real quick. Looking about these plant hormones, you know, plant hormones, if you think about it, are just elements that are made up, or not elements, they're made up of the elements. So talking about cell division, cell expansion, you know, thinking the metals, the zinc, the manganese, and the copper. Talking about sugars, the phosphorus, the potassium, magnesium, for their role in chlorophyll. How do vines take up nutrients? Really quick. Mass flow, we apply them in the water, in the transpiration stream. The water is sucked up, goes up into the canopy. The second technique is basically your diffusion from a higher concentration to a lower concentration within the roots. So basically our banded or our broadcast fertilizers are diffused, and then root interception. Here's an interesting thing here. We might, on cloudy days, so certain areas, growing conditions, growing areas that are cloudy days, mainly your uptake is by diffusion, because think about it, if you've got a 75 or an 80 degree day, you're not transpiring very much compared to that, the sun, a sunny day, you're active 80, 85, 90, 95 degrees, your active transpiration, your active mass flow for your nutrient uptake. Some of your elements are going to be taken up differently in a sunny day versus cloudy days. So if things got cloudy for 2 weeks or 3 weeks or a certain period, it's going to have an effect on the nutrients, which nutrients are taken up and which amounts. The other thing to the right is kind of an interesting thing, trying to get color, whether it's a table grape or get sugar for raisin grapes or wine grapes. But under a cool night, you see just a very consistent sugar increase, a decrease in acidity, and your flavor goes along, and then all of a sudden you got your increase in your flavors. Whereas the hot harvest nights, that bottom, sugars accumulate differently, not to the same extent. Acidity drops faster, but our flavors don't ever get to that same intensity. And so much different in how grapes perform. Berries are interesting. So up until veraison, and we showed it in some of those other charts, the xylem, the berry is getting fed by the xylem, not much from the phloem, but you see that phloem curve from veraison on. Now the phloem, that redistribution of photosynthase, is now pushing back down onto that berry. And so understanding the role of how we fertigate, how we foliar, and what the vine wants to do naturally, and how we want to work with it to get what we want. I am about done. I'm just going to click to a couple things fast. Basically, we want to do canopy management. You know, you got to balance your crop, your canopy, leaf size, shape, color, balance our micronutrients, macronutrients. And remember this, you're growing this year's crop and you're also growing next year's crop. So you've got these basal buds right there. And for several years, I actually did leaf or bud analysis under a microscope and sitting in a little cubicle all day long in the wintertime. But if you look at those little that little primary cluster is going to look like a string of pearls, and that'll be next year's cluster. So as you fertilize this year, if you're short on your elements, you're going to be short on that. One, the presence of a flower bud or a flower cluster, but two, the size and the shape. And so we're always farming multiple years. And that's it.
Yeah.
Is any questions,
Thanks,
John?
Dave. Thanks, Dave. That was some great information. There are a couple questions. Couple of them are in regards to foliar calcium. So kind of a two-part question. What type of foliar calcium do you prefer? And then any organic foliar calcium. Calcium was the second part of that question. So
yeah, I'm gonna be generic in the first answer. There's a lot of good foliar calcium products on the market today. Most reputable manufacturers, whether they're there's just a lot out there. Yara has some, most all of your big chemical companies. Organic calcium, that's going to be a little more. I know there's a few organic calciums that are out there. I guess talk to Miranda. I didn't go to the OFAC meeting the other day, but there are organic calciums. So just talk to your supplier. I'm gonna say they're all good, and then some people might have preferences, and that's fine. There's clear products, there's suspension products. Both have time and a place. So.
all right. Thanks for that. And then the second two questions, one's from Jim, the other one's from Daniel, are in regards to sampling. So which sampling position provides the most accurate level of nutrients in the tissue, petiole, leaf, or sap?
You know, I've kind of got out of the sap. It kind of started after I did most of my deal. So I'm not an expert on the sap. Most of the time I always pulled leaves and petioles, like I say, and ran them separately and analyzed them separately, and then look at them, because you might have, say, 2% potassium in one and 1% potassium in the other. And you're like, wait a second, if I just made a decision on one, I'm deficient. If I made a decision on the other one, I'm sufficient. But yeah, the best would be to do a right at bloom, and that's where the standards are made. Now, if you want to do your own standards, pull them when you want, but do the same time every year. And so you establish your own standards, knowing that generalized curve that I glossed over, nitrogen's always going to start high, calcium's always going to start low, magnesium's going to start low. So if you're not feeding certain elements, you're just counting on the soil with that transpiration stream to get those elements up. And so, and then either at a veraison or at a harvest is the next. So I would say sample at least 2 or 3 times a year, set your own standards, and take it from there.
Perfect. Thanks, Dave. And that kind of covered the second question as well. So again, thanks, Dave. Thanks for your time. We're going to move on to our second speaker, Sebastian Korob. He's a crop manager with Yara North America. Sebastian, thanks for being here, and the floor is yours. Thanks again, Dave.
Thank you, John. Thank you, Miranda, for the opportunity. I assume you are seeing my full screen, so if that's not the case, please let me know. So over the next 20-25 minutes, I'm going to talk about processing tomato crop nutrient management. And I'm going to be focusing on the work that we have been doing in the last couple of years and share with you some of the research data. The focus of the presentation and really the focus of the work that we have been doing is toward identifying opportunities to increase productivity and profitability when it comes to crop nutrition. So In particular, what I'm gonna discuss today is crop nutrition applied to 3 specific nutrients that are critical for tomatoes. They are nitrogen, potassium, and calcium. And I'm gonna be talking around the approach that uses the 4R of nutrient stewardship to build crop nutrition programs. So I'm gonna be focusing particularly in nutrient requirements to define the rate of application, distribution of nutrients throughout the growing season, to define the right time of application and different forms of nutrients and fertilizers, to talk about the right source of fertilizer to use. And at the end, I'm gonna share with you some of the research data coming of the use of this approach. Starting point to define a crop nutrition program is to define the plant requirements. Different crops in different geographies will have a different requirement, but we can still use some general information, different literature that is available to establish the nutrient uptake of a pound per pound of a ton of fruit. So if we look at this chart and we focus on the work done by Dr. Hart, we see that there are specific recommendations in terms of pounds of nutrient per ton of tomatoes for nitrogen, phosphorus, potassium, and so on. And if we look at a specific crop, for example, a 60-ton crop, then we can use those recommendations to establish the general amount of nutrients that will be required in order to achieve that particular yield. So for a 60-ton crop, we are looking at nitrogen 180 units, elemental phosphorus 27, elemental potassium 240 units, calcium 120, and magnesium 36 units. Of course, that we need to look at these as plant requirements. That doesn't necessarily mean that is the amount of nutrients that are going to be applied. That will have to be determined by looking at specific field conditions, environmental conditions, soil type, soil fertility, even the previous crop, the variety that is being used, the irrigation system, the irrigation water, the nutrients that are coming in the irrigation water. So all those factors will have to be taken into account when it comes to establish the final target for nutrient application in order to meet those plant requirements. Another key point is the distribution of nutrients throughout the growing season. So this is going to help us to define the right time of application. The nutrient uptake curves, in this case here for nitrogen, help us to identify the nutrient demand throughout the growing season. So if we analyze that nutrient uptake curve with more detail, we can target specific amount of that nitrogen budget for different moments during the growing season. So if we look at what is happening during those first 30 days after planting, there is a low demand of nitrogen. About 20% of the total nitrogen is required during that period of time. And if we look at the following month, going from 30 days after planting to 60 days after planting, 55% of the requirements for nitrogen are needed for that particular period of time. So what that tells us is that during that month, the second month after planting, and of course that this is going to change depending on the variety and the geography, right? This is something that we need to keep in mind when analyzing the nutrient uptake curves that are not particularly adjusted for every single variety and every single geography. So as a general guideline, what this tells us is that during that period of time is when we want to make sure that most of the nitrogen is available for the crop for uptake, because the requirements are very high. If we look at a crop that is 110 days, that's really a short period of time. It's during that early bloom, fruit set, and early fruit development that most of the nutrient requirements are happening. And we are talking about nitrogen here, but that is almost something that we can translate to the rest of the nutrients. And then the big portion when it comes to time of this crop that is in the ground from day number 60 to 110, only 25% of the nitrogen is required. And again, that doesn't necessarily mean that that nitrogen will come from the nitrogen that we are applying, right? It could be significant amount of nitrogen that are coming from residual nitrogen in the soil that are available at that period of time, and that is going to also affect the availability. And in the case of nitrogen, that particular stage is important because if more than required of nitrogen is available, that could conduce to some plant quality issues. Now moving from nitrogen to potassium, we also see that there is a potassium uptake curve that shows us the distribution of nutrients and their requirements. Similar to what we said with nitrogen, there is a period that goes from day number 30 to 60 when 50% of the potassium is required by the plant. But then in this case, the fruit development stages going from day 60 to 110, there is also a significant amount of potassium that is required during that period of time. And that is explained by the role that potassium has in the fruit, the amount of potassium that is uptaken in the fruit and that is removed with the plant in the fruit as well. And when it comes to calcium, there is not a graph here to share with you, but this chart is showing us that there is a more consistent distribution of requirements for calcium throughout the growing season. It's not very different from early stages to development stages to maturity stages, the requirements. So that is telling us a lot about the plan that we need to put in place when it comes to making sure that there is enough calcium available throughout the season. Another key component of the fertilizer program is the right source of nutrient and the right source of fertilizer. And when it comes to nitrogen, we know that there are three forms of nitrogen in the fertilizer: ammonium, urea, and nitrate nitrogen. Ammonium and urea nitrogen are converted to nitrate for uptake by the plant in the soil, and that's a process that happens at different rates based off moisture conditions and temperature. So it's important to look at the different fertilizer groups because the nitrogen form that prevail in that type of fertilizer is gonna tell us a lot about the availability of that nitrogen, especially right after application, before that process of conversion from urea to ammonium to nitrate is happening. So we have fertilizer sources that are ammonium forming fertilizers such as urea. We have fertilizers that are nitrate fertilizers as potassium nitrate, calcium nitrate, where the majority of the nitrogen, if not 100%, is available in the nitrate form. And we have combination fertilizers such as ammonium nitrate, calcium ammonium nitrate, where we see different forms of nitrogen in different percentages. So when comparing nitrogen sources, we know that nitrate nitrogen is the preferred source of nitrogen for tomatoes. Tomatoes is a crop and that was something that I just showed with the nutrient uptake curve that grows very fast in a very short period of time, and nitrate nitrogen is the source of nitrogen that will be directly available for crop uptake under different conditions in that short period of time. Nitrate nitrogen also promotes the uptake of critical nutrients such as calcium, magnesium, and potassium, and also increases fruit quality, especially when it comes to fruit firmness, fruit color, and shelf life. Also, there is research that shows that nitrate nitrogen reduces the susceptibility of certain diseases, in particular fusarium, which is a disease that could be quite detrimental in our geography. And part of the research that we are doing right now is focusing on understanding the value that we can get by using programs that have more nitrate nitrogen to minimize the incidence of fusarium. In the last couple of years, we conducted research trials in growers' fields. And one of the things that we did is to install lysimeters in the ground at 3 different depths. So there were lysimeters installed at 8 inches, where the majority of the root density is. Lysimeters at 16, where we see a decreased level of root density, and at 24 inches, where almost no root density exists, or I should say roots that are actively uptaking nutrients at that depth. And then we compare different programs. One program that will have a balanced amount of the 3 different sources of nitrogen, and another program that will provide mostly nitrate nitrogen, which is the form of nitrogen that is immediately available for crop uptake. And then we measure the amount of urea, ammonium, and nitrate that we found in the lysimeters at the 3 different depths. And what we found is that the fertilizer program with higher content of urea and ammonium resulted in larger levels of nitrogen losses. So what we can see here on the chart is that is represented as the nitrate level, the blue bar, at 24 inches. So that program that has a balanced amount of the 3 forms of nitrogen ended up showing a higher amount of nitrate levels deeper in the ground at levels where the roots are not actively uptaking nitrogen. So what that means is that the leaching, the nitrogen losses that we experience in that case were higher. So what we concluded is that mainly the soluble urea was moving down in the soil profile with water, and then it was converted into nitrate nitrogen deeper in the soil profile, and that was away from the roots. At the end of the day, we want to make sure that a more effective fertilizer program that keeps the nutrients at the right level and in the right place is providing us with better results. And when we compare those 2 programs, we observed an increase in productivity of 31% with the program that was showing a higher use of nutrients, or I should say a more effective and efficient use of nutrients. And when it comes to nitrogen in particular, it shows here an increase from 42% to 75% in terms of the nitrogen use efficiency. Now moving into calcium, they already talked about the role of calcium. Calcium is a critical nutrient, especially for fruit crops, when it comes to the role that it has in the cell wall and the cell membrane, and the key role in assuring fruit quality. calcium moves with the transpiration stream, and this is very important to understand some of the physiological disorders in tomatoes, such as blossom and rot, that are caused by calcium, and how we need to fertigate our crops in order to avoid for that to happen. And there are different ways to apply calcium. Calcium could be applied with soil amendments, but we know that continuous supply of calcium with fertigation is the way that guarantee that the soluble calcium is going to be available when the plant needs it. And to that point, talking about fertilizer sources, we know that there are sources of calcium that are used in amendments like gypsum and lime that are not necessarily soluble as a calcium source for the crop. So when it comes to deciding which calcium source is used in the program, we need to make sure that is a calcium source that is soluble, that will be available when the plant needs it, such as calcium nitrate. There is extensive amount of research done in Yara and elsewhere that shows that continuous supply of calcium reduces the incidence of blossom and rot, and also improves overall fruit quality measured in terms of firmness and shelf life. In that chart and the graph, what we see is that increasing levels of ammonium nitrogen, in this case applied as ammonium nitrate, over calcium nitrogen increases the level of blossom and rot. So by assuring that our program has continuous application of calcium, in this case with calcium nitrate, we reduce the level of blossom and rot and therefore increase the marketable fruit at harvest. And speaking to calcium, is important also to talk about soil quality, and soil salinity is a significant issue in our soils, and it affects plant growth and development. The fields that we were working with in 2021 showed very variable levels of salinity, with EC going from less than 1 to close to 10. And what we observe is that some fertilizer programs that use more calcium, and not only calcium, but in general, more positively charged nutrients, such as calcium and potassium, would lower the level of salts, especially when that salt is sodium in the soil, because that sodium is being removed from the soil particles and replaced by calcium. Now moving into potassium. Potassium is a key nutrient for tomatoes. It's a nutrient that is needed in large amounts. Is needed even in larger amount than nitrogen. And most of the potassium, like I said earlier, is exported with the fruit. And one of the most impactful, if we are focusing on productivity and profitability, one of the most impactful roles of potassium is the potassium role in assuring color uniformity and even ripening of tomatoes. The deduction that growers can experience from green fruit at harvest can be quite significant, and potassium is a nutrient that can influence that percentage of green fruit at harvest. And ultimately, potassium has an impact in yields that is contributed by the potassium that comes in the program in pre-plant applications and during the growing season as well. I think potassium availability could be a common topic of discussion in our area. Most of the field where we have been conducting trials show low levels of potassium. And then when we do tissue analysis in CEU, that shows as well that those fields with low levels of potassium in the soil and fields that don't receive an application of potassium that meets the requirements will end up showing also low levels of potassium in the tissue as well. But what is interesting to see here is that regular soil test that, according to most labs, will provide a sufficiency level of a level that is in between the sufficiency range. When we do analysis more in detail and try to understand how much of that potassium in the soil is soluble and available for the plant, whether we are doing a saturated paste test or we are installing lysimeters at different depth and measuring the soil solution, the level of potassium, we find that that soluble potassium is below the sufficiency range. So what that tell us is that if the program is not including more potassium, then that potassium that is going to become from that soil during the growing season is not going to be sufficient. The programs are very variable. We work with growers throughout the area, and there are growers that are not using any potassium at all, and there are programs that will go with potassium up to 200 or more units per acre. Again, it's a very field by field type of decision. I think what it's important to understand is that there is a plant requirement of potassium that is high, that our levels of potassium in the soil are, for the most part, not sufficient. And whenever we increase the level of potassium in the fertilizer program, what we are seeing in early analysis of our data is that there is a correlation with the level of potassium in the plant tissue, and ultimately a correlation with higher productivity and better quality of the tomato crop. And here, wrapping up, I just want to share with you 4 different fertilizer programs that include different fertilizers products recommended at critical stages. There are different degrees of intensity of the different principles that I just presented in these programs. And at the end of the day, what it's important to understand is that we need to evaluate the level of nutrients coming from each one of those fertilizer programs. So in particular, when we talked about these 3 nutrients, by assessing more in detail the products that are being applied and when they are being applied, we can differentiate the nitrogen in the form of nitrate and the nitrogen in the form of ammonium in this program. We can separate out of these programs, or wherever is the program that is being applied, the actual amount of calcium that is being used, and the same thing with potassium. When we look in particular at these 4 programs, going from a baseline program all the way to a program that adds more value, we see that there is an increasing level of nitrate, nitrogen, there is an increasing level of calcium, and there is an increasing level of potassium as we move through those programs. And at the end of the day, the transformation by focusing on the crop nutrition and key aspect of crop nutrition is about implementing a comprehensive approach that help us to increase the productivity, the quality, and the profitability. And we know that in California, we are seeing a decreased number of acres that are going to tomatoes. It's very variable year to year, depending on the market. But if we talk about overall, for what we see in the last number of years, that number of acres is decreasing. But at the same time, the levels of productivity that are needed by the processing plants are still high. So if we look at implementing a comprehensive program that increases productivity in 15%, which is the average level of productivity increase that we are seeing when we implement this type of approach, then we are looking at a lower number of acres in California that could be needed in order to meet that same allocation. And that is significant when it comes to the future of the tomato industry in California. And last but not least, the importance of creating an increased profitability for the grower, right? There are agronomic principles. We see an increase in productivity, an increase in quality, but if we don't add value to the grower, if we add value to the crop nutrition program, that needs to come with a positive return on investment for the grower. And that's what we are experiencing over the last 6 years in the research that we have conducted. And with that, I am done, John. I'm not sure if there is any question.
Yeah, thanks for that, Sebastian. I know processing tomatoes has been a little bit of a hot topic or hot crop, looking into 2023 just because of shortages and such. So I hope some of you CCAs out there, learn something from Sebastian's presentation and consider incorporating it into your programs next year. There is one question from Daniel. He's asking to go back and he wants to reference slide 19 again, if that's possible, real quick.
Yeah, this information is available, so we can discuss more in detail with advisors if they want step-by-step type of explanation with the programs. I think what is important here is not just to copy and paste the products and the amounts that are recommended, because that needs to be adjusted field to field. Is to understand that those principles that we have been discussing are implemented in order to build this program, adjusting the nutrient demand with the fertilizer that is being applied at the right time in the right amount, looking at the different sources of fertilizer. All those concepts are included here in order to build this program. So if any particular advisor wants to sit down and review this program and even brainstorm with some of the programs that they are using these days, I'm happy to do that.
Perfect. Thanks for that, Sebastian. Yeah, and contact information will be shared here in the chat box at the end of the webinar. So again, thanks, Sebastian. We will I guess we'll move on to our third speaker, last and never least, if you know Devin. Devin Clarke is our permanent crop manager, Yara North America. Devin, thanks for taking the time on Friday afternoon. The floor is yours.
All right, thank you very much. So I'm going to go ahead and pull this up here and get this into presentation mode. It's always tough following somebody as technically savvy as Sebastian here, and as much information as Dave's years of work in the industry have been able to provide. So I think we should be in full screen mode now. But
there we go.
Yeah, came through. Okay. Perfect. Yeah. So I tend to kind of take a bit more of a kind of a storytelling approach. So as John mentioned, I am Devin Clarke, permanent crop manager, primarily work here on the West Coast and Western region, and focusing primarily on nut crops, almonds, walnuts, pistachios, as well as doing some work in wine grapes and avocados these days. So what I wanted to talk about today was really the journey to establishing a clear value proposition for soil health. It's a very challenging one, and one that is has a lot of work yet to be done, and very specific work in individual cropping systems to be able to fully derive a value proposition. So I wanted to start out with just a quick question on what is considered a healthy soil. And so with that, we can look at 3 different images here. We see kind of a forest floor. It looks like on image A, looks like a compacted salt-affected soil here in image B, and maybe a grassland type of setting in image C. So of these 3, which one actually would be considered a healthy soil? And with that, the answer is it depends, right? The purely political answer or academic answer, I would say, is it depends, right? And it depends on the perspective that you are evaluating this from. So are we talking about natural versus working lands? Are we talking about annual versus perennial crop systems? Are we looking at this from the perspective of the grower or the consumer perspective, who is asking for, more health, more sustainably produced food and more healthy food? And are we looking at this from the perspective of a Midwest soil? This is one of the bigger ones, the regional parameters. Is this a Midwest soil? Is this a California Central Valley soil? And even within there, we know and recognize that we have a substantial amount of variability within soils in California. So, and I do apologize for any of those on the call today that are going to be at the Alman conference, because I will be doing this presentation again. So this is just a prelude to that. But I wanted to talk about specifically within almond production systems, what are some of the opportunities that exist, and also what are some of the challenges? So when we first talk about soil health, we have to understand kind of what is the framework that is currently established for quantifying or qualifying, we'll say, soil health. And in this image here, basically the 3 areas that are focused on consistently and are really critical to any discussion on soil health are looking at the physical, the chemical, and the biological aspects. And Dave actually pulled up a slide earlier in his presentation that captured that too, and had some more definitions that fell under each one of those categories. But the nice thing about this framework is that this can be applied and adaptable to any cropping system, any soil type, and any location. But there's these indirect and these direct interactions that we have to understand. And really we have to be focused on what is the outcome that we are striving to achieve. And that's going to really frame how we develop a management strategy to attack the concept of soil health. So within almond production systems itself, I think that, for one, the infrastructure that we have from an input delivery standpoint, right? If you're talking about drip, micro sprinklers, even subsurface drip is beginning to show up a little bit more in the industry these days. We have a really unique opportunity compared to other cropping systems to be able to precisely deliver our resources in terms of water, crop nutrition inputs, and even soil amendments in some cases, right? Or microbial food sources. So that capability really gives us a unique opportunity to really put what we want exactly where we want it and be able to make significant changes in those areas around that root zone. The other one I would say is the fact that this can be both detrimental and positive, and I'll cover the detrimental side in the next slide. But the orchard lifespan, right? So many of these permanent crop systems, in particular to almonds, they are going to be in the soil for around 25 years, give or take, right? So that permanence that exists there, along with the fact that we have essentially already created a no-till system, aside from the planting processes, gives us that a unique opportunity to potentially build some characteristics around soil health. Additionally, the root architecture and just the biomass. So not only the way that the roots are structured and that architecture of branching with primary, secondary, and kind of tertiary roots, we have really a great opportunity to impact a large volume of soil with the spacings that we have with these orchards, with the amount of root biomass that occurs below ground. It gives us that opportunity to produce those carbohydrates in the upper parts of the canopy and move those down through into the soil and feed a lot of these soil microorganisms. That branching structure also gives us the ability to mine a great degree of volume of that soil and provide access to things like nutrients and water to that plant, and simultaneously then create that feedback loop again that help support carbohydrate production in the leaves and the canopy and move that back down to the soil biomass. And also has implications in soil structure when we can use those exudates to start binding soil particles together to create stable aggregates. And then the last one I would say would be biomass recycling. So there's kind of a newer push towards whole orchard recycling. That would be one consideration in terms of biomass recycling that is able to basically incorporate a large amount of carbon and all the compounds that exist within the almond tree back into the soil. But also throughout the course of every season, we have all the leaf matter that ends up back to the floor. We have all the prunings that take place and end up usually being reincorporated back into the soil to get chipped up and then reincorporated back into the rows. So there's that opportunity to reintroduce that carbon component time and time again through just the recycling of the biomass that's produced in the almond cropping system. Now, there are considerable challenges, I would say, within almond production systems that hamper efforts towards improving soil health. But some of the key ones that I wanted to focus on would be primarily water quality and availability. That's obviously a big one at this point in time. Harvest management practices, the fact that almonds, different from say pistachios in this case, we shake those to the ground and then we sweep them up. And that process in and of itself means that we need to have a very clean orchard floor in a lot of cases. And so we're removing all of the biomass as much as possible as we near that harvest time so that we have the ability to gain higher efficiencies out of our pickup equipment, reduce the dust production in the process because we don't have to set the heads all the way down to the ground and start stirring up a lot of dust in that process. But this practice of shaking these nuts to the ground require that we clean a lot of that vegetation off, and maintaining vegetation throughout the growing season is a really key step to improving soil health. The resource competition that exists of looking at maintaining clean herbicide strips throughout the growing season, that's also another challenge that is difficult to overcome. Compaction, looking at the early parts of the season, particularly I would say in the central Valley as well, or say we'll say in this case the northern San Joaquin Valley and then up into the Sacramento Valley, being able to get into the orchard in the early part of the season to be able to apply those fungicides and apply any of those nutrients that may be required to support early development and mitigate disease pressure. That requires that we're taking tractors out in the middle of the orchard when that floor may still be wet and we're creating compaction in the process. So the last one that I would mention would be the assessment factor, right? Being able to accurately assess the changes that are taking place over time. In contrast to what Sebastian and others have been able to achieve with annual crop systems, right? We've been fortunate in almonds that some of the funding has been provided to be able to create research projects that are doing full tree dissection and have been able to help produce crop uptake curves for us. But in general, to be able to really evaluate these systems, the permanence of our cropping system in almonds definitely has some challenges. And so in that, we have to have many years in most cases of data in a very particular location in a very particular way to be able to glean any meaningful insights off of that. Whereas in other cropping systems, you may be able to do many replicates within one field to be able to achieve faster results and faster learnings. So, and then I wanted to talk a little bit about getting in deeper to measuring that impact. So first and foremost, again, as I mentioned early on, there are lots of variations in the definitions of what constitutes soil health. One thing that I wanted to specifically call out here that appears to show up in many of these definitions is that we have to begin thinking about this soil and this plant growing environment as an ecosystem. And just to calibrate the understanding of where I'm going to be coming from in the rest of this talk, I spent 5.5 years as an advisor in the field as a PCA. I have my PCA and my CCA license. I work with growers directly. I am not by any stretch of the imagination advocating towards, full organic conversion or regenerative conversion or anything of that nature, but just trying to provide some practical thoughts around anyone that wants to drive down this road of improving soil health. And so trying to lay the foundation for that. So ecosystem here, again, one of those concepts or one of those elements that continuously shows up in many of these definitions. So when we're talking about this ecosystem, we're looking at this soil food web, and here we understand this cycling of nutrients and this opportunity for supporting the resiliency of our soil and the soil health. You know, there's so many different players involved in this, from the kind of the macro players being your mammals and small rodents and birds even, all the way down to your protozoa and predatory nematodes and bacteria, right? So we have to understand that where this occurs is typically going to be in your rhizosphere, the aerated parts of your soil. And so depending on what, you know, again, from a regional standpoint, if you're in a heavier clay soil up in the northern part of the state versus down south in the southern part of the state, you know, that rhizosphere is likely going to be much smaller. And so you have to understand about where these interactions are going to be taking place and where you have the ability to implement change. So with that thought of an ecosystem, you know, it's really going to require us to kind of change the way we're thinking, because we've spent so many years, really, and centuries in this case, right, trying to eliminate the variables that exist within food production. And we have done a phenomenal job, and we have really supported basically the global population growth with nutritious food over the years by doing so. And so we are really looking towards that next evolution, because we're not getting a lot of new AIs coming onto the market that are helping us to control certain pests in a very mechanical way. And so we have to recognize that there are natural systems that are in place to be able to help mitigate some of the pressures that we fight on a daily basis from a consulting standpoint. And so for this, in this slide here, I just have a few examples for navel orangeworm specifically. You know, we have parasites or parasitoids on the upper right. You can see the black one there. I'm not even going to try to pronounce the species at this point in time, but you can see all of the eggs that are hatching and growing inside of that navel orangeworm larvae on the right-hand picture, and the fact that that has a really strong ability to control navel orangeworm. We have on the bottom left, we have a fungal element. This is a Bovaria bassiana that has the ability to significantly wipe out colonies of navel orangeworm, though it's very difficult to synthesize in the lab. And I know that there is at least one commercial product in the market, but it's very expensive and it's very difficult to synthesize. Next to that, we have BT, the black worm, there on top of the almond skin. That's also a naturally occurring bacterium that exists in almond production and has the ability to wipe out navel orangeworm in very localized areas. And then lastly, looking at from a more physical standpoint, you know, there was recent research done by Houston Wilson and a number of other co-authors that investigated the ability of winter cover crops to reduce the emergence, primarily I'll focus on that, of navel orangeworm, because all of the competing or all of the different species and the level of moisture and everything that exists within the base of that cover crop basically reduce the ability for that overing winter larvae to survive. And so that can actually have a good impact on reducing navel orangeworm populations in the early part of the season. So as I mentioned, we really have to basically move from what, you know, we typically are looking at the soil surface in a lot of cases. If we're looking at the soil at all, in a lot of cases, we have a lot of folks who have spent many, many years staring up at the canopy. So turning that attention down to that soil, not looking at it from a 2D perspective, but looking at it from a 3-dimensional perspective and understanding what are the functions and supporting mechanisms and the antagonistic mechanisms within your particular production system that that you need to be aware of to be able to establish any particular framework in attacking the discussion around soil health and how you can implement management practices in your own systems to be able to overcome those. So the first thing that the one thing that I really wanted to make sure that you took away from this discussion is that because these definitions are so varied in nature and because of the regionality of the different soil types and conditions and management systems that we have, I think it's really important from an agronomist standpoint, from a grower standpoint, that you start with a very tangible and quantifiable goal. So in this particular one, I have, I want to focus on 4 different things here: the outcome-based approach, the functions that are existing within the system that can influence that outcome, the supporting elements that influence those functions, right? And then the antagonistic elements that you need to be aware of that may cause trouble to achieve your objectives. So with this particular example, let's take a grower who's lacking water availability. Let's say their water costs are about $1,000 per acre foot. For a 40-acre field, their water bill is going to be about 160,000 acres a year, or sorry, $160,000 per year. So as a very real and tangible goal, let's say this grower wants to save 10% on their water costs without compromising yield. And that's a very important factor, because we have a lot of different stakeholders that exist within the supply chain that are focusing on really absolute cuts when it comes down to climate smart farming and things of that nature, versus focusing on the actual units of, say, carbon dioxide production, CO2 production per unit of food produced, right? That's a really important distinction that we are very adamant about. And so that's why I wanted to highlight this without the compromising yield factor. So if that's the case and this grower wants to save 10% on their water costs without compromising yield, you're looking at about a $400 an acre savings goal, or potentially 4.8 inches of water being better utilized to be able to produce a better crop. And so when we look at this goal, let's start with the functions and think about, okay, what are the areas within my cropping system, the systems that really support this water savings goal? And so we have to think about water holding capacity. We have to think about the root access to be able to access that water within the soil. And we have to think about how efficiently that tree is utilizing that water and turning that into crop at the end of the day. Now, the supporting elements that can help get you to that goal are going to be focusing on soil organic matter, soil structure, irrigation scheduling, infiltration rate, crop nutrition management, right? Those are some very key goals, or some very key areas to be focusing on. And so this really helps you kind of narrow down your focus, as opposed to trying to have this very broad approach to a very specific problem. And then additionally, again, we have to be cognizant of the antagonistic elements like soil water chemistry, soil texture that is very difficult to change, distribution uniformity of our irrigation systems, and again, that compaction factor that is very difficult to bypass in a lot of cases. So this all comes back to the old adage that you can't measure what you can't or you can't manage what you can't measure, right? And so we'll get a little bit more granular into some of those measurements that support those focus areas. So I'm going to be highlighting a little bit of information that we have been able to gather over the past year that came from our Better Soil Alliance work. So we established this Better Soil Alliance in conjunction with Hele Agriculture last year. We brought on some tech partners to support our learnings. And so this is just kind of a brief overview of what it is that we're trying to achieve. So we're trying to basically activate a community within the almond market to drive practical solutions that positively impact food production by improving water use efficiency, soil health, and lowering greenhouse gas emissions. So I won't get too deep into this one right now, but please, come, reach out to us or reach out to us at the Almond Conference if you want to learn a little bit more. But the one area that I did want to focus on here is the protocols, is that we do have established protocols, particularly revolving around the utility of calcium nitrate and Hele's Phycoterra product to be able to support improvements in the chemical, the structural, and the biological components of soil health. And the next step is really that the rapid assessment that we have in the bottom middle column. And so we've established this 3-tiered structure for evaluation that helps to overcome that long lag time of being able to generate learnings from permanent crop systems that I mentioned previously. And so we're utilizing our incubator farm that we have in Modesto, California as a extremely granular evaluation level. On the opposite end of that, we have what we are calling our crowdsource farms, where we are working with growers to implement these protocols, and we're working in basically a grower desk audit type of format, where we're not doing any actual measurements on site, but we're requesting certain information to be sent to us from these growers. And then in between those, we have our case study farms. So we're shooting for 50,000 acres in total. Our case study farms are intended to make up about 10,000 acres in total. And then we have our incubator farm. But this helps us not only, like at the case study level, I forgot to mention, we do have these Phytech sensors, we do have series, we do have additional resources to help us evaluate on-site changes with physical sampling efforts and physical monitoring efforts. That is also in addition to what we have at the incubator farm. So these three tiers basically give us the ability to not only reach scale, but also to be able to provide granular level learnings of what we're achieving at the local levels. So with that being said, some of the things that we are measuring very specifically with the Better Soil Alliance protocols is we're looking at aggregate stability on the wet aggregate stability side. There's not a lot of labs that are doing dry aggregate stability at this time. We're looking at water holding capacity. From the chemical standpoint, we're looking at the soil pH, all the nutrient profiles. We're looking at EC, cation exchange, base saturation, sodium absorption ratio. And then on the biological side, we're looking at specifically organic matter, active carbon, PLFA analysis, which is a very extensive microbial DNA kind of analysis that provides us visibility on what types of species and what types of diversity we're seeing in these zones. And then also ultimately you see at the bottom here, crop yield is a key component. And then beyond that, we're also looking at the nitrogen and the water inputs and looking at the efficiency values that are generated in these protocols. So from an incubator farm perspective, I want you to kind of think about that previous example we had where the grower had this 10% savings goal, $480 an acre, or I think it was about 5 acre inches, and it will actually pop up here again. But what we've been able to achieve on average over the past 3 years is that we have the water treatments are equal across all of these 4 programs. We do have 6 replicates of each within a 27 acre section of an almond field. The only difference between these is the inputs and the way that we're managing them. So the My Almonds program that has produced the best results over the last number of years is working off of a high frequency, low volume approach, coupled with a balanced and timely nutrition program, essentially built around the 4R structure. Now, this water productivity is the graph here is basically the way to read this is you're looking at the number of applied acre inches of water per unit of kernel yield. So that's that per unit of food production that I talked about before. And so we've been able to, only by changing the inputs and changing the timing of those inputs, nothing with regard to the water or the infrastructure, we've been able to achieve a 14.7% increase on average over the last 3 years. And so for each year, that equates to about 7 acre inches of water, or $580 an acre using this previous example. And we've been able to do that with a 3-year average net ROI of over $1,200 per acre. So when we take that back again into context of that previous example, it was $1,000 an acre foot, $160,000 a year, want to save 10%. And we're looking at $400 an acre or 4.8 inches of water. We were able to exceed that by almost double in case of the water. And we were able to achieve $580 an acre on average over those 3 years. Now, the Better Soil Alliance protocols were only rolled into the My Almonds program in the last year. And so here are some of the soil health metrics that we looked at over the past year where we saw some decent differences. And so we were able to identify some modest improvement trends against the baseline in the active carbon, in the total microbial biomass, and then also within the organic matter. So these are multiple replicates again, and we are evaluating this over the course of the growing season. We're starting in basically April and running all the way up until October. And so we have to understand, again, the regional context. This is a very light textured soil. And so the opportunity to make vast improvements over the course of one growing season in terms of organic matter and carbon and things of that nature and water holding capacity, we have to understand that naturally those are very low, right? But we do have to begin investigating how these things play out in different scenarios and start to build a portfolio of information to be able to then start driving management decisions. So when we get down to trying to really quantify that value, it's really important that from a grower and an advisor standpoint that we understand what the trade-offs are. This is not comprehensive by any means, but I wanted to highlight a few areas that as you are working with growers, or if you are a grower yourself, these are the kind of things that from a very practical standpoint, you have to take into consideration when wanting to make specific improvements in the area of soil health. So think about if we increased our organic matter and you were relying on that organic matter to supply some of your nitrogen. We understand and recognize that the greatest demand for nitrogen occurs in the early part of the season for almonds. And so with that, we also have lower soil temperatures, which reduce microbial activity and reduce the mineralization of that organic matter. And so then some of that release of that nitrogen from that organic matter may take place in later part of the seasons where it may be less advantageous or yet less utilized. And so we have to understand that this increase in and of itself, has to be calibrated against the predictability of the supply of nitrogen relative to what the crop demand is. When we talk about increasing soil carbon, we also have to recognize that, as any advisor that has ever had a faulty application of herbicides and has had to try to come in and utilize an activated carbon or something of that nature to try and tie that active ingredient up to minimize any further damage, this increase in soil carbon is also going to tie up and increase the sorption of that herbicidal active ingredient to that carbon, and it's going to make it less available in that soil system, making it less available to do its job in a lot of cases. And so that's a very real and practical consideration. Greater water holding capacity. Again, as we talked about that in that early season compaction issue of having to get in the field to apply fungicides and nutrients, right? If we increase that water holding capacity and get greater infiltration and greater holding capacity, that there are very real implications about being able to have access to the field in the early part of the season and what the actual compaction that's going to occur. So we're almost taking one step forward, two steps back in some cases. And then lastly, greater above ground biodiversity. Again, we touched on it earlier, but thinking about the increased resource competition, and some of those things are very difficult to quantify. So one thing that I know that there's been so many instances of taking kind of a Midwest example and trying to apply it to a permanent crop situation that we've seen in the almond industry over the years. This is not intended to be that, but simply just a call out to how to be able to potentially look at, from a budgeting standpoint, being able to quantify the improved ROI to the grower by making specific practice changes and using this as a template to look at this partial budget analysis so that you can explicitly quantify that value from a grower ROI standpoint. Your categories just may look a little bit differently than what they have in this particular example. So in conclusion, I just wanted to highlight again that from a soil health improvement standpoint, there is no one size fits all approach. There's very specific regional, temporal, and site-specific context that is required in every single situation, whether you're talking about trying to improve carbon, whether you're trying to incorporate cover crops, whatever that is, you have to understand the site-specific context and what those trade-offs are. There are some very unique opportunities within almond production systems that provide us a great starting point to be able to make some of these improvements. But again, we have to have a very tangible goal in mind. And there are numerous indicators, but there are no specific formulas. And again, just touching back into that no one size fits all approach. So again, we want to start with very practical and measurable goals so that we can be very targeted in our efforts and make sure that we're measuring things in a fashion that are going to give us real results and not pseudo results. So start with an outcome in mind. think about the soil functions that are going to support that, look at the supporting elements that feed those soil functions, and be aware of those antagonistic elements. And then lastly, again, look at those trade-offs, understand what those trade-offs are relative to your goals, understand what the impacts are to your total system, find out where, what elements are really non-starters for you in the way you want to manage and the way you're set up to manage, and then which ones can be overcome. And really take the time to provide that economic analysis so that you can really thoroughly quantify the value, so that you don't have those situations where a grower is willing to take that leap of faith with you to try and go down a path of better soil health, and then you don't have the ability to really quantify that value. That is where we lose a lot of people in the process, because you just don't have the ability to directly show how this is going to benefit you long term if you continue down this pathway. And with that, I believe I have got myself wrapped up for the day.
Thanks, Devin. Definitely appreciate it. Soil health is definitely a topic on the minds of most of us on the ground, in the agricultural industry, but it's also reached the consumer, and it's been further driven by major food distributors like Pepsi and Costco, Blue Diamond, Wonderful, et cetera, et cetera. So much appreciated. There is one question, but Devin, I'll put you and Jim in contact individually just to be cognizant of time. So okay. Miranda, if you don't mind putting up that QR code, we will we'll have things wrapped up here. So there's a QR code. As you know, once you scan it, you should immediately see the hours in your hours reports. So please go ahead and scan. In the chat, we've put the speaker's contact information, so feel free to give them a holler if you have further questions. Of course, I want to thank the speakers right on behalf of Western Region CCA. Definitely appreciate you guys donating your time. And then, of course, thanks to all the attendees for attending the second installment of the Western Region CCA fall webinar series. Really hope you found the content valuable, and please come back for the third installment, which is on December 16th, same time. So outside of that, thanks again. Have a great day, evening, fantastic weekend, and Merry Christmas. Thanks again.
Miranda, can you slide your screen up?
Yeah, there you go. It's getting covered by the bottom bar of Zoom.
So I think that's
the QR code's getting cut off by the bottom bar of Zoom.
Okay, let me. So on mine, it's fine. Does adjusting the size fix it for everyone?
Yeah, that worked.
Oh, maybe mine's still getting cut off. Maybe you just shoot it over to us afterwards, if you don't mind.
Yeah,
I got it.
Yeah, it worked for me.
If it didn't work for you, please reach out to us, to Miranda directly, and we'll get you that QR code.
Oh, there it is. I'm good. Thank you. Thank you, Miranda. Thank you all.
yeah, no problem. Everyone have, like John
Thanks,
said, have a
everybody.
good evening and a great weekend.
Yep.
Thanks, everybody.
Take care.
Thanks, John.
Thanks, Miranda.
No problem. I'm just gonna leave this up here for like another couple minutes and take it down at probably like 3:11. So if I know sometimes people have issues with the app or their phone in general. So
All right, cool. I'm gonna jump. I gotta grab my daughter, so I will appreciate it. I'll talk to you soon.
okay, thanks, John. Have a good one.
Okay, bye.